So Insilico Medicine decided to spin out the longevity business, and all of our aging-related research is currently with the Deep Longevity. It's a new company that we announced like a couple of weeks ago, and I'm leading the science there, so I'm a chief scientist at Deep Longevity. And today I will present our deep biomarkers of human aging. That the work that we've been doing while still being part of Insilico, but now it's with Deep Longevity. So just to put you in perspective, a bit overview of the company, because it just newly started. We are an AI company focused on biomarker development. We founded in 2019. We just recently closed our Series A, received funding from some of the longevity investors that Aubrey mentioned during his talk. And our main specialty is aging clocks, in particular deep aging clocks. So we use deep learning. This is sort of next generation artificial intelligence to predict biological age of individuals. And up to date, we have a broad portfolio of those aging clocks. Some of them are patented. We tend to publish as much as possible. So we have many peer-reviewed papers on those. And currently we're developing the digital platform, which we call Young AI. So the aim of this platform is actually to integrate our aging clocks and allow people to access them and to use them. So a bit of an overview, what is happening right now in the biomarkers of aging. I understand that Professor Steve Horvath just gave an amazing talk on epigenetic aging clocks, but there are many of those that has been published so far. Of course, with his seminal work, everything started in 2014 when he published his epigenetic clocks, and those advances in generation of those biomarker of aging was mainly driven by availability of new data types, so epigenetic profiles, transcriptome data, and so on. But there is another revolution that's happening right now. It's also a revolution in terms of algorithms. So we have a new technology that called deep learning that started to revolutionize image recognition, self-driving cars, voice recognition, and so on in 2014. And since 2019, it's actually changing the healthcare. And most of the hype around the technology is due to really credible advances in deep learning and reinforcement learning. I know that some people are still skeptical about what is happening there, but you just have to see it to believe it. And why it's happening right now? Well, mainly because we have new algorithms, so we have a way to train them. We have computing power, so we can actually train them. So it's not only theoretical work that started a long time ago, but it's something that we can actually do computationally. And we have a lot of data to train them because they're exceptionally data hungry. And regulatory actually recognize the potential of deep learning. And since 2017, Food and Drug Administration, US, approved more than 14 AI-based tools. And for most of them, FDA granted fast track approval. So because they're exceptionally accurate in the way how they perform those tasks. So those tools mainly are not aimed to replace doctors in any ways, just aim to assist doctors with their routine tasks. Like for example, the Arteris Cardio recently approved MRI heart recognition engine by FDA, and or Apple iWatch that they got their approval on the atrial fibrillation detection in September 2019. So it's happening. So the deep learning is there, and it's changing the healthcare, and it's changing dramatically. So what is happening right now with biomarkers of aging? So since 2016, multiple deep learning based clocks has been proposed and published. We also contribute a lot to this list. And right now, some of those clocks are only available for deep learning based algorithms, like for example, the facial image recognition or bone X-ray clock or brain MRI clocks. They only, or deep microbiome clock that we developed, they are only were possible to be made just using deep learning. Of course, all of them vary in accuracy in terms of age prediction. And this is the same as for the standard machine learning approaches for predicting age using transcriptome or epigenetic profiles. And all of them have their pros and cons. So why do we actually have to have a lot of aging clocks? Apparently, they measure different things. So this is an amazing study by Professor Dan Belsky on comparison of 11 biomarkers of aging. So he compared several epigenetic clocks with the telomere, with also the other blood-based markers of aging. He predicted the biological age of people with the same chronological age. And he showed that while they are same chronological age, they have a significant variation in their prediction in terms of biological age, predicted even by different methylation clocks. And while all of those clocks are working the same way, so if we analyze longitudinal data, they're ticking in the right direction unless we intervene. So people are getting older by their telomere biological age or methylation age. At the same time, they apparently predict different aging processes. So that's why we are focused on developing a really diverse portfolio of aging clocks, aiming to capture as many of those processes as possible. So we work with the really different data types, starting from the omics data, like transcriptome profiles, microbiome profiles, and going to facial images or data that we get from wearable devices. And the first clock I will present today is the deep hematological clocks. This actually was the first one that we published. I have to say that first that blood markers, so those simple blood parameters like albumin, glucose that we use to predict age, are not markers of aging by themselves. Of course, we can see them changing, let's say, in the right direction. So we know that, for example, albumin tend to decrease with age, or glucose and cholesterol, total cholesterol, they tend to increase with age. At the same time, the correlation is not strong. And it's not like if you will be given a number of albumin levels or cholesterol levels, you will be able to tell how old the person is. So that's why we actually have a model that can combine those parameters and capture the dependencies between changes in parameters level with age. So to do this, we used an ensemble of deep neural networks. So it was a combination of 21 different deep neural networks with completely different architectures, meaning they have different hyperparameter set, different number of layers, and so on. And we trained them to predict age of healthy individuals. After that, we of course validated on the prediction on the test set that was previously unseen by the model. But to aggregate all of those predictions, we also used the second model. In this case, it was like a simple elastic net model to predict age. And to further validate the model, we also decided to predict sex, because obviously blood parameters are sex specific. That's why we have different ranges. There are sex-specific ranges, like for example, creatinine or for hemoglobin, but it was a proof of concept for us as well. So when we received first results, we saw that we're able to predict age of healthy individuals with 5.5 mean absolute error. And while those people were healthy, we still saw a variation there in predictions. So some of them were predicted older than they actually are, and some of them were predicted younger. And I will later show you why it's really important to analyze those cases further to understand what is happening with the model. Then we decided to see what was really important for the model to assign the age. And those top features, of course, included albumin, glucose, and other parameters that we know change with age, but those individual parameters are still not able to predict age of individual accurately. At the same time, right now we also working not only with the importance of those parameters in terms of age prediction, but also direction of change. So for example, if you increase the level of albumin, then the sample that were predicted older will get younger, and other way around for glucose. So for example, if you increase the level of glucose, then the person will get an older blood age, as we call it. So we're currently playing with those algorithms to see whether we can actually achieve an optimal blood levels for individual, so they will get younger. After that, we decided to see whether we can actually validate our clocks on different populations. So the previous model that I showed you was trained on Eastern Europeans, and we then went further and collected a really substantial data set for Canadians and South Koreans to validate the model. And the way we designed the experiment was that we trained several age predictors. So some of them were trained on individual populations, and one was trained on the combined population. So it was like a sort of universal one. What we saw was really interesting when we started to cross-test them. We saw that if model was trained on only single population, is actually tend to mispredict individuals from other populations. So while all those people were healthy, if model was trained on Eastern Europeans, for example, it tend to predict Koreans significantly younger their chronological age, and other way around. So if model was trained on South Koreans, it tend to predict Eastern Europeans older. So having a combined model seems to solve the problem. And a combined model is actually even more accurate in predicting age of individual populations than just a model that was trained on those individual populations, which was really interesting to see. So there's something in there, those population differences, that helps model to assign age. Then we decided to see, all right, would the most important parameters, the list of most important parameters will be different for different populations. And apparently there is difference between Canadians, Europeans, and South Koreans. Yet those 4 common criminals, they're still there. So albumin, glucose, hemoglobin, urea. So they're the most important parameters across all populations in predicting age. Then we decided to analyze those cases that I showed you previously when people were predicted older or younger than they are. So we did the mortality analysis, and we showed that individuals who were predicted older, their chronological age, they had a higher hazard ratio and higher mortality rate, lower life expectancy compared to people who were predicted younger. So depending on population times, those associations were statistically significant. So in this way, we show that we show that the age we predict using the blood profiles is it's a better predictor of mortality than just a simple chronological age or single chronological age by itself. So this way we show that what we build here is actually a deep aging clock, because it's associated with mortality and with lifespan. We also show that those clocks are able to reflect lifestyle choices. So here's an example for tobacco smoking. We first trained a model on 23 parameters only on non-smokers, again, healthy non-smokers. Checked if the list of parameters, if the list of the most important parameters for model predictions is the same. And then we added smoking status as one of the factors for predicting age. So model actually were able to predict age more accurately in this way. The list of the most important parameters is still almost the same as was before, but then we decided to analyze, all right, what if we will test smokers and non-smokers in terms of biological age prediction to see whether there are any differences there. And we saw that smokers, of course, were predicted twice as old compared to non-smokers. But at the same time, we also saw an interesting effect here that it's only happening for the people who are younger than 50 years old. So you will see no difference, and even for the people who are older, and even for the cohort of 80 years old, they are predicted sometimes younger. So, well, that's, if we go to the literature, we see that there are several potential explanations there. First of all, the smoking by itself, it could be one of those so-called like protective factors. For example, nicotine by itself, it's considered to be neuroprotector, and it's currently explored in terms of its ability to prevent Parkinson's disease and other neurological conditions. At the same time, smoking is also could be like a stress, sort of a stress factor that helps people to boost their health and somehow prepare for the aging changes. At the same time, we also believe that when you're already like over 6 years old, probably smoking is not one of those leading factors that contribute to you getting older. So there are other, especially internal factors that will drive this process, and regardless if you're a smoker or not, you will see no difference in terms of blood age. We also were able to predict the smoking status by itself using just simple blood parameters. So there definitely a huge difference in terms of blood profiles of smokers and non-smokers in that regard. And again, that's another illustration of those results. So if you analyze non-smokers and versus smokers, you will see that they are predicted significantly older by all age groups at the same time. So another clock that we developed together with those deep hematological clocks, as they call them, is tissue-specific transcriptomic clocks. That was a paper that we published in 2018 in Frontiers. The way we did this experiment is that we collected a profile of healthy tissues from public domain on skeletal muscle profiles. And then we decided to actually compare different algorithms here. So we compared deep neural networks with other standard models, including the elastic net regression here, and to see whether there will be any differences in terms of accuracy with age prediction, but also in terms of features that those models consider to be important for age prediction. We also proposed it as one of the tools how we can rank genes by their importance and then explore them as further prospective dark targets. So it wasn't surprising to see that deep neural network outperform other models. Actually, a support vector machine was also one of those models that performed almost with the same accuracy as a deep neural network. But what was interesting to see when we analyzed those ranks of importance of each individual genes assigned the different models here, both deep neural networks and also shallow models. We combined them to obtain the final rank. And then we saw that the deep neural networks actually were able to obtain the ranking really close to the final rank. So from the beginning, deep neural network in the end kind of had a ranking close to the consensus ranking of all other algorithms. So that's again another motivation to not only go for shallow models, but also for a deep one, because it's apparently, let's say, more consistent across different samples. Another type of clocks we are exploring as well is a deep microbiome clock. We managed to publish this paper quite recently. That was actually our collaboration with Harvard University, with Professor Vadim Galashev. What we did here, we collected microbiome profiles, whole genome sequencing. Then we analyze them in the way that we used abundance profiles of each of individual microbe and use them as input vectors to the algorithms. Of course, we always compare deep neural networks with other models. And here we show that we can predict age with the mean absolute error of 5.9 years. But what is also interesting to see that when we analyze the people with type 1 diabetes versus healthy controls, we saw that people, like individuals with diabetes, they are predicted older compared to healthy controls. Of course, those clocks are still lacking the validation in terms of the mortality prediction, but microbiome profiling by itself is rather a new field, so we don't have the data yet for that, but we are really looking forward. So if anyone is actually interested in this type of collaboration, we would really love to collaborate to see whether we can actually validate this type of clock on mortality data. Then with this deep microbiome clock, what we also can do, we can also see again the contribution of each individual microbe taxa to their age prediction. So the importance in terms of age prediction, but also what we'll do if we will change those abundance profile. So what we'll do, what will happen with the predicted age if we increase or decrease those particular individual microbes. So will the profile will get younger or older? This way we simulated thousands of changes and come up with the so-called seropositive and seronegative status of those species, meaning that seropositive, those were species that while we increased the abundance, the predicted age also increased, and seronegative, while we decreased the abundance of those profiles, the age also decreased. At the same time, when we decided to analyze whether there any association with their function in the body, we saw that there is some association, but not much. And for example, some of those pathogens, they were actually associated with the decreased age prediction, just because they are known to be associated with the youth infections. So that's why we decided to come up with this classification here outlined on the slide, when we also introduce whether they are pathogenic or beneficial to the body, and then class them by seronegative and seropositive status. All right, so just to sum up, again, there is a revolution in aging clocks happening right now that was mainly driven by the accessibility of different data types, but because of the revolution in deep learning and AI in healthcare, we see right now a lot of clocks developed, those deep learning aging clocks. And using those clocks, we can not only predict the age, biological age of individual, we can also identify what kind of features out there are associated with the age the most. And then later, as I showed for microbiome profiles, we can also simulate what will happen if we will change them, and would it be possible for us to identify those optimal profiles that will actually make individuals younger, at least for deep neural networks. Of course, it still has to be tested in terms of interventions will actually benefit the individuals, but that's something that we are working on hardly right now. So thank you for the attention, and I'm happy to take any questions there might be.